Hello A2, AS. Unfortunately I can't be with you today. I've been lucky enough to be invited on a training session, so which wasn't confirmed until later, so apologies about that. However, you're not going to be able to get away with it completely because I'm going to talk you through the lesson instead, as well as obviously you've got Stuart for cover. So these are the things that you're going to be looking at today. You are going to be doing an some exam question marking as always, and then you'll get feedback from this YouTube video. You will list as many evaluation points as possible for experimental methods. You will make notes on the nervous system, the blank diagram, and see what you have missed. Check your learning. You will complete the nervous system mobile that you started there at the front of the table, and at the end when you finish with them, can you put them back into the little white folder that they came from, please? Create a revision resource for the types of neurons, and I'll show you on Padlet and I will talk you through the instructions as well. Do a cut and stick activity of the synaptic transmission, and then check the answers against the YouTube video. There's an extension which is a very detailed and complex video of the synapses focusing on summation, which I would encourage you to watch, but only if you're really keen to stretch yourself quite significantly. The worst thing that you could do is watch it and really panic and worry. So if you're thinking that you're a little bit unsure, that's absolutely fine, then go on to the next activity. However, if you finish all of that, you feel 100% confident, then I would recommend that you do watch it and see how you go. If you don't get all of it but you get 10% of it, that's brilliant. So that's another option. And you can start completing the exam questions on Moodle and the flipped learning. So there's absolutely loads for you to do. No one should run out of anything. The first thing that you are going to do, and this might take a while to start moving through, is I want you to find the contextualizing activity. I'd like you to read through the sheet. They should be on your tables in front of you. If not, Stuart or whoever it is that's covering will have a copy. So I just want you to pause the video, go get yourself a sheet, read through it, read through every bit of it, and then I would like you to give it a mark, so what you think it is out of 3. There is one on there that doesn't have a mark, which is question 3. Where is it? There we go. C, it is out of 4. So for all of them, I want you to look, tell me whether it's got a mark, full marks or not. If it hasn't got full marks, tell me why, and tell me how you would get it full marks. If it has got full marks, tell me why, and then don't worry about adding any other information. So go get that worksheet, pause the video, complete it, and then start it again, and we will go through the next, the answers. Okay, so we've done all the to-do. Hopefully you've answered all the questions on the sheet, and then if we go through the answers, so question 1, hopefully you noticed that it was not repeated measures, it is independent groups on the sheet. The design and evaluation points do not match up, of course, because it's not repeated measures, it's independent groups, and the strength is for repeated measures, so you do still you don't suffer from order effects in the independent groups. However, hopefully the really PD-eyed amongst you will notice that you won't get any marks because it's not repeated measures and independent groups are not experimental methods, they are experimental design. I've put this one in, and I'm going to put it in a few times if I remember in the future, because it's consistently a mistake that students make. Do not talk about the experimental design if it asks about a method. Experimental design, repeated measures, independent groups, matched pairs. Experimental method, lab, field, natural, quasi or quasi. Okay. So they won't get any marks for that. You need to tell me which type of method you think it is. It's a lab, and then a strength or a weakness of a lab, because I think it has to evaluate it, and don't forget to contextualize it as well. So make sure, is it a lab? Have you said that it's high in controls, easy replicable, whatever it might be. Question 2 gets 1 mark because how does having the number allocate them into each condition? So if you walk it through step by step, you put the numbers in the hat and you make sure there's an even number of both, which you've done brilliant. Then what you do is you give everyone a number. They all pick a number out of a hat, and that's it. That's no good at all. You then need to say condition number 1 takes to condition 1 and number 2 takes to condition 2. The whole sentence, the whole question is something like explain how you would use random allocation. So it doesn't have to be contextualized. However, I would rather you over contextualize than under contextualize. So do make sure you contextualize. The next question gets no marks. Why? Because it is not a relevant variable to be matched on. Does shoe size have any bearing on memory and recall? No, absolutely not. So instead we'd have something like memory memory, age, familiarity with the location or the classroom. You get 1 mark for saying how they would be matched. So you would do a baseline memory test and then match them with good on the most similar scores. Same with IQ. You find out how old they are. You'd match them on similar ages, and you'd find out how familiar they were with the classroom, how many times they've been to the classroom before. That would get you 1 mark. The second mark would say each person does one condition. So you'd say that one person does the similar in the same context and one does it in a different context, and that would be how you get those 2 marks. Question 3, you get 1 mark for matched pairs, but you lose 3 marks for not contextualizing. So they would have seen the photo already. So what I've done with this one here is you've done the generic evaluation points, which you could take out and put into any exam question. So if the question was about repeated measures, evaluating repeated measures and why it wasn't suitable, you could put that in any question, it wouldn't matter. So because I've talked about order effects and demand characteristics, you could pick out order effects and you would explain order effects. You could pick out demand characteristics and explain demand characteristics, which is exactly what I've done here. So they would see the photo already, so would give the same answers, although they would work out that the aim was to see the impact of a leading question and purposely allow themselves to be influenced by the question. So they've seen the photo already, so would give the same answer. So it's order effect, practice effect, they're better in the second condition. Or they'd work out the aim was the impact of the leading question and purposely allow themselves to be influenced by the question. Demand characteristics, they would be purposely influenced. The next question was full marks. Why was it full marks? It used the PE structure, so point, explain, and it was contextualized, so they linked it back to the study, which is of course how all of your answers will go. 4 marks, question 4, no marks, not contextualised. And you would only, even if it was contextualised, only get 2 marks because it asks for advantage as. So please underline, circle, highlight that S. If it asks for advantages, so plural, then you need to give more than 1. If you don't give more than 1, you are limited to 2 marks. And there's one, a strength of the lab is highly controlled, extraneous variables could be minimised. In this experiment, outside factors such as waiting time, noise and stress were removed. Another strength is easy to replicate, the same doctors, facts and diagrams. So you've contextualised the same doctors, the same facts and the same diagrams. This is a strength because it would mean the results can be checked for reliability to see if we would get the same recall of facts. So that's why you've got the marks there. Right, pause this video after I've told you what to do next. So you've done that one activity, keep that sheet safe because you need to return to it. In your teams you have 2 minutes to name as many evaluation points of experimental methods as possible. So lab, field, natural, quasi, not designs, methods. I'm going to, what I found I was giving people was more like 3 minutes. So 3 minutes. If somebody else has finished at the same time, you can work with them. If no one else is finished and you want to work on your own, you're welcome to do it that way as well, although working in a team would be easier. I have 15, and I suppose you could have longer than 2 minutes. What you can't do is check your notes. I have 15 evaluation points. So when across all 4. Some of them are similar, so you can duplicate them. If you want to say a strength of a lab is that it's highly controlled, and a strength of a quasi is highly controlled, that's absolutely fine. You can have 2 of those. List as many as you possibly can on the whiteboards, which are should be on the tables. Don't waste paper, so go get yourself a whiteboard. And then come back and check this video, tick off how many you got right, add any that you missed, and then of course you can check with me if there's any that I haven't got that you've included, because they might be accurate. The one I encourage you to avoid is about ethics. There are natural experiments, there's something to say about ethics. Other than that, there is nothing to do with ethics really, lab, field, natural, quasi, unless you are linking to it's a very, very specific study. Okay, so pause the video, give yourself 2 minutes, list as many as you can, off you go. Okay, so they're the examples that I have provided. So lab was controlled, easy to replicate. I'm not going to go through them. What I did with the other classes, I'm going to do with you. I want you to pause. I want you to look through the on the slides, look through the information on there, tick up everyone you've got. Any that you've missed, make a note of them, and remember, you must remember them. And any that I haven't thought of, save them and tell me about them, or you can email me and let them know what they are, especially if they're good ones, because I obviously haven't thought. So pause it here and check them. Okay, next thing that you are going to do is you're going to recap biopsychology. So on your tables there should be this lovely, which you've already seen before, diagram of the nervous system, like so. I would like you to complete the A3 sheet, which is printed on A4, so it is only an A4 one. No notes, nothing, and the absolute minimum amount of time I'm going to give you to do that is 2 minutes. For 2 minutes, no matter what else you're going to do, whether you can write something or not, I want you to sit there and include as much as you can. If it takes you longer than 2 minutes, that's brilliant. Keep writing. The minimum with no notes is 2 minutes. After that, if you need longer, you just keep writing, include anything that you can. After that, go to your notes, check your notes, and add anything in a different coloured pen, and identify one particular thing you think, right, next time Harriet asks me to do this, which will happen again, then this is the thing that I will include on my nervous system. Okay, so have a go at that, and then we'll move on to the next activity. So pause me here. Okay. The next one is several tasks all together, and you can choose the order as long as they all completed, or at least very, very close to completion. I would like you to, oh dear, the picture covers that up. Complete your mobile. So think about the mobiles that you began at the start of the session there at the front of the classroom. I want you to come and collect them, and I want you to finish them off. And what's being covered by the lovely, the beautiful photo is that you must include visual imagery. So think about the occipital lobe. There's an eyeball on there. I'm not bothered what the imagery looks like. I just want it. So if you're not very artistic, it doesn't matter. Neither am I. This is where creative thinking comes in. You want to think about how you can draw it as simply as possible, because really it's about cueing your memory, so reminding you of those things. So I want you to finish your mobiles, create some visual clues, possibly on the back add a bit more detailed information. You decide. After that, 1 second, I'm just going to sort this slide out. There we go, much better. So, the first task, complete your mobiles including visual imagery, and then you are going to place the cut and stick activities in order. Don't stick them down yet. They will go on the back of this worksheet here. So if you have a look at this worksheet here, go right onto the last page. It says neurotransmission cut and stick activity. That is where you are going to include this information. Okay, so where is it? There it is. There we go. I'll make them a bit bigger in a minute because I want you to see them. So place the cut and stick activities in order. The cut and stick activity looks like this. I want you to cut them and place them in the order. So this is for synaptic transmission. So this is where we talked about things moving from one neuron to another. And I want you to do the best that you can with this. We are going to do it again next week, but I really want you to have a sound grounding, because this will be the third. By the time I meet with you, it'll be the third or fourth time that we've covered it. Okay. So have a go at placing that in order, and in a moment we will go through that order. The next thing I want you to do is create a diagram of each type of neuron: sensory, motor and relay. There's some crafting stuff at the front of the classroom. What I want you to do is create a revision resource, and as you can see from these excellent examples here, what they have done and what I would like you to do is you create the image using as much or as little crafting activity as you want. Don't get too hung up. It's great to have really beautiful images, and you can take them away with you later on, but what I don't want you to do is get too caught up on it and then miss the point. What you need to be able to do is name the neurons. So I'd like you to put a label, name it, and then cover the name with a post-it note that says name ? The idea is it's a bit like what I like to call a revision flap. So you lift it up, and underneath is the name of the thing that you are that your arrow is drawn to, and then the role as well. So this one here, my guess is if it goes to here that it's the cell body or the nucleus. So you'd lift that up, you'd check our cell body. What's the role of the cell body? Well, the role of the cell body is to hold the nucleus and hold the genetic material for the rest for the neuron. Okay, and we've done it for all of them. You can see exactly the same. This one is one individual one, and that's fine, or you can do all three together. And if you finish before the time is up, I'd like you to do all three. If you only manage to get one completed, then that's fine. So you've got the neurons here, and then you've got the role, ? Name ? And so on. And you can revise by lifting up the flaps and saying what it is. Okay. The other thing you can do is all of last year's ASs. So if you want some idea on Padlet, these 3 Padlet walls. So if you follow those links, you can have a look on the Padlet walls. There's some really, really excellent and very creative ideas for that one there. Okay. So work through all of those things for me, please. and then come back. One thing I'm going to share with you at the end of the video is the homework, so if you want to make a note of that in your diary or something, your phone, you do need to get that out and make a note of that as well. So all these three things must be, two things must be completed. The third one you must be very, very close to completing. If you complete both of those, you can start getting on with the flipped learning for next week and your exam questions for next week, and I'll talk you through those in a minute, so you can get a real head start. So it's absolutely beneficial for you to be focused and complete everything, bearing in mind we're going to go over it next week, and I will assume that you already know something about it, so make sure you also fully understand it. So pause, work through those. The next thing I'm going to go through on the video is the answer to is the neurotransmission, order of the neurotransmission. If at any point you get stuck on that, you can go through my video. You can look at the 2-minute if you Google YouTube 2-minute neuroscience synaptic transmission, or go on Moodle. There is a link to the 2-minute video as well as my video, which explains it all as well. Okay. Right, so if we go through the answers to the neurotransmission card sort, where you can stick them down, you've got the nerve impulse or action potential runs down the axon of the presynaptic neuron. We've been through all of this on the video before. Then the nerve impulse reaches the terminal buttons at the end of the presynaptic neuron. This triggers the vesicles to move to the edge of the membrane and release the neurotransmitters. They are released into the gap, they travel across the gap, they bind to the receptors on the postsynaptic neuron, and then this is summation. Depending on the type of neurotransmitter, the postsynaptic neuron can receive either an excitatory or an inhibitory action potential. So we're talking about inhibitory or excitatory summation. The neurotransmitters are released by the receptors and returned back to the synaptic gap. The neurotransmitters are reabsorbed by the presynaptic neuron transporters in a process called reuptake, and the vesicles are refilled with neurotransmitters. If you're ever not sure of this one, a tip I should have given you before the answers is highlight the the words pre and post, and not always, because obviously down here you have got presynaptic neuron matching, but most of the time pre is at the top, post is at the bottom. So you can stick those down, and then the final thing I think I've got to talk you through, yeah, because I've done all that, is your homework. So let's have a quick look. Your homework is this here. I would like you, as always, so none of this is new, okay? Complete the exam questions on Moodle. So there's the synaptic transmission ones. Mark the exam questions. Do the flip learning in the approaches booklets. So the approaches booklets should be on the table. They're the big thick booklets. You're going to be working through them for 6 weeks, so don't panic. And they look like... Come on, computer. It'll come up in a second. I will show you what the approaches booklets look like in a minute, but they will be on your tables. You complete page 1 and 2, so I have effectively remembered to number them, which makes it helpful. Really small amount of flip learning. There's the A3 sheets that I gave you last week. You want to be filling those in, going through those. And then the key thing I would like you to do as well is a revise for an assessment, which will be next week. That has been on the Google Doc, the homework Google Docs for a while. If you want to check when the next assessments are coming up, you can have a look on Moodle. And it says a homework deadline sheet, and it tells you all about when the assessments are coming up. Andy's potentially changing. He doesn't edit that, so they might just keep it listening to him, but I have put the original dates on, and obviously I should edit mine as they go along as well. But you can check on there. Things that you'll be doing, so in the 1 hour session with me, so that's Monday or Wednesday depending on which group you are, hypotheses, percentages, primary data, measures of dispersions, controls, extraneous variables, and everything to do with biopsychology. My advice to you would be to complete the exam questions on neurons and synaptic transmissions before the day that you have to revise, before the day that you have to do the assessment. That's all I'm going to say. It will help you a great deal if you have already seen a mark scheme. Right, let's have a look. The approaches booklet, which is taking forever to load up, you won't be able to see it, is, let's see if I can get to it this way. It will be on the table. It's a big booklet that has lots of different images. There we go, that one. And the only pages you need to complete are that one about Wundt and introspection, and the emergence of psychology as a science. do not go on to the behaviourist approach. Well, if actually the videos aren't up yet, but if I manage to upload some videos and you want to go into the behaviourist approach, you can do. The only 2 pages you need to have ready for next week are Wundt and page 1, Wundt, and the emergence of psychology as a science. Okay. Any questions, please do email me. I will be checking my emails, or you can ask Stuart and he will leave a note for me as well. I hope you've enjoyed your virtual lesson for the day. I'm sure you miss listening to me on YouTube, and I will be back with you next week. Please make sure you get the homework in. Thank you.